Hello, everyone. I'm so honored to come your way again. So we're going to be doing up a follow-up questions following the last video that I had made regarding understanding ABO discrepancy. Okay, so I had already mentioned to us previously that this question came from someone who had wanted me to clarify or speak on the possible factors that could lead to some of the ABO discrepancy. And the person gave me an image which I had shown us the other time, which I'm still going to show again today. Then I had answered the first one. So today we are going to look at the second one. So before I continue, my name is Dr. Emmanuel Obodo. I'm a specialist biomedical scientist, a senior biomedical scientist, and I'm HCPC registered, a fellow of Institute of IBMS, and of course, I am a visiting lecturer here in the United Kingdom. Just in case if you do want any consultation, please feel free to contact toogoodconsultant@gmail.com, or you can send me a WhatsApp message using the number shown on the screen. So yeah, let's get into this. So like I did mention, the person had shown us this. So I have done the first part, which is the first one, which is this patient one. I'd already finished that. So we're going to look at the patient two. So just as a quick recap, if you remember what I had said, when it comes to ABO discrepancy, simply put, that anytime the forward did not agree with the reverse, or the reverse did not agree with the forward, that is ABO discrepancy. What it technically means is that, like I'd mentioned to us before, when we look at the forward group, we are measuring the antigens on the red blood cells. And when we look at the reverse grouping or typing, we are measuring the antibody in the plasma. And I did mention, don't forget that the antigens are found on the cells and the antibodies are mainly found in the plasma or serum, okay? So when we look at ABO discrepancy, there are two ways you have to look at it. First, you are looking at immunoglobulin M, antigen A or antigen B, or immunoglobulin M, antibody A or antibody B. So most of the things I'd already explained on my previous video. So I'm going to quickly move into this. But before we continue, just in case if you've not subscribed to this YouTube channel, please feel free to subscribe and make sure you like, share, and comment. So let's look at the patient number 2. So this patient 2, you are going to see from this patient 2, the forward group said that it is positive, strong positive, by the way. It is a strong positive on anti-A. Anti-A, I had already explained this on my previous video. So because it is strong positive on anti-A, that obviously shows us that this patient is A positive, okay? Now, it's A positive because if you look at anti-B, the patient on the number 2 is negative. So it's negative on anti-B, that's anti-sera B. And when you come to anti-sera A and B, it is positive, but weakly positive. So depending on how you look at it, but it's also a confirmation that this patient is A positive. So I will tell you why. So when a patient is blood group A, the person has antigen A and antibody B. So we'll come to the antibody later, but for now, the patient has antigen A. So if you look at this place, on this anti-sera A, it is embedded with commercially prepared anti-sera, that means antibody A. And if you look at anti-B, it's also commercially prepared embedded anti-B. And this one is commercially prepared embedded antibody A and B. So what that means is that because this patient has antigen A, that antigen A reacted with the anti-A, anti-sera A here on the anti-A well. And of course, because the anti-A and B well contain antibody A and antibody B, so I believe that this +2 is a reaction of the patient red cells antigen A reacting with that antibody A that is here, okay, not the antibody B. So the forward group agree that this patient is A positive, even though I would have expected this to be stronger than this. But for the presentation, for the purpose of this presentation, this shows that this patient is blood group A. However, there is a problem. Remember that if someone is blood group A, the person should have antibody B. That means the person should have antigen A and antibody B. So I'd already explained all of this on my previous video, but just like a quick recap. So when you look at this forward group, is using commercially prepared antibody to test what is on the patient red cells, which is the antigen the patient has got, whether antigen A or antigen B or no antigen at all. Now, if you look at the reverse grouping, it's using a known A cells, a known B cells, a known O cells, and of course the patient own cells to test what is on the patient plasma, okay, because that is where the antibody will be. So that means the reverse group, we are using the cell to test against the patient plasma where the antibody supposed to be. So the forward group had already showed us that this patient is blood group A, but the reverse group is saying otherwise. So let's look at it this way. If you look at the reverse grouping, it's showing that under A cell, it is weak positive, that means +2. Under B cells is strong positive, that means +4. And under the O cells, okay, it is weak positive, which is +2. Now, auto is negative, which obviously is the kind of result we want. That means that it is there is no evidence of auto antibody, or as the case might be. So now, if you look at this, there's a challenge here, because if this patient, which is blood group A, has antibody B, that antibody B, it makes sense why it reacts with the B cells, because antigen has to be specific to its antibody. So that do make sense. And this also suggests that this person is blood group A. Why? Because if you look at the B cells, it's strongly positive, which is +4, compared to the positive on A cells and O cell. However, even though the A cell and O cell shows a weakly positive, that shouldn't be there in the first place. Remember that A cell can only be positive if the person is blood group B, because blood group B will have antigen B and antibody A. So that antibody A is what are supposed to react here, but not on this occasion that it is blood group A, which means it has antigen A and then it has antibody B. So this reaction here on A cells and this reaction here on O cell becomes something for us to ask a question. Why should that be the case? And don't forget as well, which is also another surprising thing here, it is surprising to see a result on the O cell. Why? Because O cell should not have antigen A, should not have antigen B. So technically, it shouldn't really react to anything. That O cell should always be negative because it does not have A cell. It does not have A antigen or B antigen. So there shouldn't really be anything technically. There shouldn't really be anything technically for it to react to it. However, we are seeing a weak reaction on A cells and a weak reaction on O cell, and obviously strong reaction on B cell. Now, I told us the other time that when it comes to ABO grouping, and of course in blood banking, one of the things that can help you to make a decision is the degree of the reaction. So for the fact you have seen +4, it confirms what is there, that it is more reliable than the ones that are weak. It doesn't mean that we're going to relegate the ones that are weak, but it helps us to understand why, to investigate why we should actually have that weak reaction in the first place. So if you look at this, you will see that under this patient too, there's a strong reaction on the forward group when you look at anti-A, and there's a strong reaction on the reverse group when you look at B cell. So which obviously confirm that this patient is blood group A, meaning it has antigen A and antibody B. So the next question we now need to ask ourselves, why are we getting a weak reaction on A cell, and why are we getting a weak reaction on B cell? Okay, so don't forget, if you've not subscribed to this YouTube channel, please do subscribe. Okay, now let us look at what could be the possibility of this kind of result that we've gotten. Okay, let me stop you there, okay? Now, I want you to do me a favor, because the statistics of this YouTube channel suggest that many people who are watching this YouTube channel, they are yet to subscribe, okay? Please do us a favor. Do me a favor, subscribe if you have not done so, and don't forget to share, like, and comment. That way, it can take this video to many people that this video will help. So together, we can help colleagues to be able to locate this video and get well prepared when preparing for their what? Their interview in biomedical sciences, or as the case may be. Also, I wrote a book, okay? And I believe that this book will help you if you are preparing for your interview. I want to kindly ask you to go on Amazon and get this book. I'm also going to put a description on this YouTube channel so you can, directly purchase from Amazon. Okay, let's get back to the video. Okay, now let's look at this now. So if you look at this patient 2 now, you will see that there is obvious thing here. So I had already put it on the table just to recap what we have seen on the previous slide. So we already know that there's a strong reaction here. You can see +4 on anti-A. Then you can see +4 on the B cell. This obviously confirms that this patient is blood group A. However, there's a problem, okay? I'd already mentioned that this weak reaction we have on +2 here, which is under anti-A and B, I would have expected it to be stronger, but for this presentation, I'm going to say, okay, that is still fine, because it can be argued that it is reacting to the anti-A here, not the B, and maybe the competition there, as the case may be, is what I give it that very weak reaction, okay? But I would have expected it to be stronger. But let's not worry so much about it. The one that is actually imposed challenges on this very ABO discrepancy is the A cell, the one I highlighted with blue color, light blue, A cells and O cells. So A cells can only be positive if the patient is blood group B, like I'd mentioned, and this O cell should always be negative because O cell does not have antigen A and does not have antigen B, therefore cannot react with anti-A or anti-B as the case may be. So that is the main issues here. And of course, the autoantibody, meaning that the cell of the patient tested against the patient plasma is negative, which is fine. What this does to us, it helps us to rule out that these very weak reactions we are seeing that is causing this ABO discrepancy, that it is not autoantibody, which is very, very fine. Now, I had put my explanation here just as a recap, just to make this things easy for us, okay? So we're going to look at the possible causes, okay? Number one, I want us to look at this from what I'd already explained. You will see that the forward group confirmed that this patient is blood group A. And obviously, there is no autoantibody. So because there's no autoantibody, we now know that all these the reactions we are seeing in unexpected wells are not due to autoantibody. Now, if you look at the reverse group, now the question, remember, when we do reverse group, well, even though we call it A cells, what we are using is A1 cells because of the percentage of the people that have, or population that has A1 cells are many, much many compared to the significant low amount of the individual that are A2 cells, okay? Now, so the reason why we may have this weak reaction here is possible, if possible. So this unexpected A1 cell reaction could be the possibility of maybe if it is A2 antigen. That means if we are suspecting that this patient, although is blood group A, is A2, okay? It is A2 subgroup of blood group A, if that makes sense. So if you look at people who are blood group A, so the main group, the greater population are under A1 cells, but there are some that are A2 cells, I think some about A3 cells as the case may be, okay? So now the only way we can have this weak reaction on A cells is if there's a possibility that we're looking at A2 antigen and not A1 antigen. Maybe that would explain that, okay? Even though I don't think that is the problem here. That's why I put query, is a question mark. Is it possible that this patient is A2 subgroup, okay? Now, Of course, we expected a reaction on B cell, which is fine, so it's not a problem. Then there is unexpected reaction with O cells, like I've mentioned. That shouldn't really be the case. Why? Because O cell does not have any antigen A or antigen B, so that shouldn't really create a reaction there. Now, having said that, what then could then be the problem? The problem could be because of antibody against known ABO antigens. So now this reaction we are seeing, don't forget, before I continue, don't forget that the plasma is where antibodies are. So if you are seeing discrepancy, you are seeing on A cells or you are seeing on O cells, you should be looking at the possibility of antibody that are interfering on this. Okay, so now what that means then is the fact that there's a possibility that there is an antibody that is against known ABO antigens. And these antibodies could be something like cold reactive antibodies, something like maybe anti-H or anti-I or auto-antibodies. But remember, from our presentation so far, you can see that we are going to rule out auto-antibodies. Does that make sense? Okay, we're going to rule out auto-antibodies because the auto is actually negative. But what could have caused this ABO discrepancy? The reaction we are seeing on A cells, the reaction we are seeing on O cell could be due to antibodies that are against, that can react to any ABO antigens. Okay, so those antibodies could be perhaps what we are seeing here, and maybe that is why it is weakly expressed. Okay, now because most of these antibodies, like in a case of cold reactive antibodies, they are mostly weakly expressed, and that could be an explanation of why we are getting a reaction on A cells and O cells. So that could be due to cold reactive antibodies. Okay, now it could be something like maybe anti-H or anti-I, and like I've said, it could have been auto antibody, but because this patient is auto is negative, the chances are is not. So now, what are my conclusion about the possible causes why we should have a weak reaction on A cells, a weak reaction on O cells, which has led to this ABO discrepancy? Number one, it could be the fact that this patient is a subgroup of A2 cells. So I've told you that blood group A can be, most populations are A1, most people are A1 cells, okay, but some can be A2, some can be A3. So there's a possibility that this patient is A2 cells. And if this patient is A2 cells, the chances are the antibody may be specific to the A cells. Remember, this antibody, this A cells here you're seeing is actually A1. Ideally, I'm writing it based on how the person gave it to me, but ideally, this should be A1 cells. So if that is a A1 cell and the person is A2, the chances are the person's antibody, not because the person is not A1, that is these cells. Now, the person being A2, possibly the antibody may be reacting with these very A1 cells. That is the likely cause. But another cause could be due to cold reactive antibody, as I just mentioned here, maybe anti-H or anti-I, as the case may be. That could be the antibody interfering to this. So in my conclusion, I would say that this patient is blood group A. Okay, this patient is blood group A. Why? There is a strong reaction in anti-A on the forward grouping, and there's a strong reaction of +4 in reverse grouping of B cell, which is +4. So that confirms that this patient is blood group A. However, the weak reaction we are seeing in A1 cells, and the weak reaction we are seeing in O cell could be due to could be due to cold reactive antibodies like anti-H or anti-I, as the case may be. Or it could be that this patient is not A1 cell, it could be A2 subgroup of the blood group A. Does that make sense? So now, what do you do when you encounter things like this in the lab as a biomedical scientist? Okay, having known the possible causes. Now, except if you have a special equipment to be able to investigate that, or method to investigate that. Otherwise, ideally, you should be able to send these samples to the reference laboratory. If it is in the United Kingdom, you have what we call our reference laboratory, like RUCI, okay, and which can be NHSBT or so WBS services. That means Welsh Blood Services, okay. So any of the RUCI, this reference laboratory, the sample can then be sent to them. Is when they send to them that they cannot confirm whether this patient is actually A2 subgroup or whether the reaction we are seeing is due to cold reactive antibodies. Now, there can be a number of techniques that they use to sort this out, but once you see this in the lab as a biomedical scientist, because there is no full agreement, your analyzer anyway will not be able to tablet or give you the blood group of this patient. The reason why it cannot give it to you, even though there's a strong reaction in anti-A forward grouping and a strong reaction in B cells, the analyzer cannot tell you that this person is blood group A. Why? Because of the weak reaction you are seeing in A cells and A1 cells and the weak reaction you are seeing in O cell. Because of that, that will kind of confuse the analyzer, and the analyzer will be unable to give you the group of this patient, even though from the strength of the reaction, you can be very certain that this person is blood group A. However, like I've mentioned, maybe this patient is subgroup A2, or maybe it could be cold reactive antibody interfering, thereby giving all those weak reaction, which then now leads to ABO discrepancy. I hope that makes sense. So my take on this is that this patient is blood group A. However, there can be a possibility of co-reactive antibody that have caused that very weak reaction, leading to on both A cells and O cell, thereby leading to that ABO discrepancy. And another one could be that maybe this person is A2 blood group, and maybe perhaps that could explain why there is that reaction. I hope that makes sense. Even though personally, if I'm to make my final answer, I don't think that this person is A2. I think that there's a kind of cold reactive antibodies, that are kind of against non-ABO antigens that is reacting. And I think that is what is happening here. So this person is blood group A positive. Sorry, this person is blood group A, and perhaps there is antibodies interfering, and that's why you are getting that weak reaction. I hope that makes sense. All right. Please do let me know what you think about this video. Don't forget to like, share, and subscribe. It's important that we subscribe and share so that you can support this YouTube channel to reach many people that this video can reach. Thereby, we can support colleagues to come across this video and prepare better when they are going for their interview. All right, thank you so very much. Do let me know what you think about the video, and of course put a comment. And like I've mentioned previously, if you need I will advise you to go and get a book that I wrote on how to prepare for your interview questions. I will put the link as well on the description of this very YouTube channel. Anyway, thank you so much. Otherwise, till I come back your way again, all the best. Bye now.